So I'm going to introduce you to an evolving collaboration between Ira Tabas, Christine Garcia, and myself. We bring together expertise in inflammation resolution, pulmonary fibrosis, and ARDS. And our goal is to understand basic mechanisms underlying COVID-19 pathology. So as you know, the proximate cause of death in COVID-19 is acute respiratory distress syndrome, or ARDS, in a subgroup of people who get severe pneumonia. So a few words about ARDS, that is COVID-19. ARDS is essentially a disease in which the lungs, the air spaces of the lungs, fill up with liquid. This is pulmonary edema and is evident in the chest x-ray as in the typical ground glass appearance, which typifies pulmonary edema. Now in COVID-19, this disease is particularly vicious because it's of rapid onset, happens within 5 to 10 days. The mortality is 30 to 50%, and there are no cures. And so our goal is to understand basic mechanisms to come up with cures. So ARDS is a disease of the pulmonary alveoli. And this is the part of the lung where the gas exchange occurs. Alveoli comprise something like 95% of the epithelial surface area of the lung. And so when there's disease in the alveoli, it becomes very life-threatening. So in the case of this infection, the virus, coronavirus 2, is inhaled through the conducting airway of the lung and makes its way to the alveoli where the disease happens. I would like to emphasize that although there are receptors for the virus in the airway itself, the disease, ARDS, is not a disease of the airway. ARDS is specifically a disease of the alveoli, and this is an important thing to remember when designing mouse models to study COVID-19 ARDS. And I say this advisedly because there is a mouse model available from JAX, which really addresses virus targeting in the airway and does not replicate ARDS. So we are able to image by direct intravital confocal microscopy. We can image the alveoli, and here's an example of an image. This is a single alveolus of a mouse lung. We can also do this in human lungs, and you see here the gas, the air space of the alveolus, which is dark, surrounded by the alveolar epithelium. And next to the alveolus is the blood vessel stained. The blood has been stained green here, just for contrast. And the important structure I want to point to are these two red lines you see here. The one next to the vessel is the capillary endothelium, and the one next to the air space is the alveolar epithelium. These are the critical fluid barriers that protect the air space of the lung from entry of fluid from the blood vessels. And so when the alveolus gets infected, these barriers break down, and there's a lot of fluid flow from the blood into the alveolus, causing pulmonary edema, hypoxia, and death. Now, the reason why the alveolus is tropic for the virus is because of a couple of cells, and I'll talk about that for a minute. So this is another confocal view of a mouse alveolus, and here's the epithelium stained in blue. And you can see here this type 2 cell, which is a very specialized kind of epithelial cell in the alveolus, which contains vesicles that secrete surfactant. Now, surfactant secretion from the type 2 cell is a critical process that lines this mixture of phospholipids throughout the alveolar epithelium, reducing the air-liquid surface tension at that surface and thereby maintaining the stability of the alveolus. If there's infection of the type 2 cell, surfactant secretion is likely to be abrogated. There's no surfactant, and therefore the alveolus becomes unstable and therefore highly prone to pulmonary edema. The other cell I'm going to talk about is the alveolar macrophage. Now, the macrophage has many functions, but one of the important ones that we are interested in this context is its ability to regulate inflammation resolution. And in a few minutes, I will talk a little bit about that. But now I want you to notice that the alveolus sits very snugly against the alveolar epithelium. In fact, it actually adheres to the epithelium and is thereby able to communicate signals from itself, from the macrophage to the type 2 cell. And I'll give you an example of this kind of communication in this movie, which shows a number of macrophages. So all these, the white arrows are all macrophages. This is 1, 2, 3, 4. And the dark spaces are alveoli. So the macrophages are spread across many alveoli. But here's a cell marked with a red arrow, which is a type 2 cell. Now, once the movie starts, you'll see calcium signals being generated in the macrophages, which travel from one macrophage to the other. But in doing so, it also goes into, in fact, the type 2 cell. So the type 2 cell is recruited in the signaling. So here we go. It's a repeating video, so you'll see it happen again and again. So here's the calcium spikes going from one macrophage to another. And as you can see, the calcium spikes also incorporate the type 2 cell. So just by generating calcium spikes under stress conditions, this was done under infective stress. So when the alveoli are stressed, this kind of signaling goes from the macrophage to the type 2 cell, and the significance I'll come back to in a minute. So I'd like to now say a couple of words about inflammation resolution, and I'm going to show you this. I'm showing you this a beautiful slide that I got from Ira Tabas, which summarizes this concept. So what infection does is it generates an early inflammatory response in which cells, mostly neutrophils, enter the alveolar spaces from the blood. But after a while, the monocytes start coming in. Monocytes get into the alveolar space and convert to macrophages. So this is a macrophage here. Now, these macrophages are able to generate a whole bunch of stuff like lipids, proteins, and other factors that act on a variety of cells to actively dampen inflammation. And here the emphasis is on active because these processes initiate as soon as the inflammatory process itself initiates. So the two go together. And the resolution mechanism, there are a whole bunch of them, including one that Ira works very avidly on, which is called efferocytosis, in which these macrophages pick up infected cells and thereby eliminate the infection. There are also these other mechanisms. So, so this resolution and repair mechanism is therefore extremely important, and we need to understand how this happens in the alveolus. So a few words about the receptor itself, and I probably mentioned earlier that the two tropic cells of the alveolus that are targets for the virus express a protein called the angiotensin converting enzyme type 2. So the virus targets this protein, and as a result, this interaction generates a bunch of peptides called angiotensin 1 to 7. These peptides ligate a receptor called the MAS receptor, also expressed on the macrophage, and this leads to inflammation resolution. So you can see that if the CoV-2 spike protein, which is the domain of the virus that actually interacts with the ACE2 receptor, if this protein degrades ACE2, which some data in the literature imply. So if there's loss of ACE2, then you can see that this entire process is going to be abrogated and inflammation resolution will not happen. So in other words, to summarize, therefore, we really are interested in knowing what these interactions are. So the macrophage sends a calcium signal to the type 2 cell to activate surfactant secretion. the type 2 cell sends a whole bunch of signals back to the macrophage to initiate inflammation resolution. And we need to know how this is going to happen in the context of coronavirus 2 infection. So our plans are to infect mice with the COVID-2 pseudovirus and the spike protein, which Dr. David Ho has very kindly agreed to give us. And we will study the cell-to-cell communication, as I've described, in wild-type mice. But in addition, we are very fortunate to have a mouse, the availability of a mouse developed by Christine Garcia. This is a very interesting mouse because it has a mutation in the surfactant protein A, which is expressed only in type 2 cells. And because of this mutation, the type 2 cell generates a kind of an ER stress type phenotype, which is the model, if you like, of a pre-existing condition. So it'll be interesting to know if this mouse generates a hyperinflammatory response to the virus. So with that, I'll stop, and I guess there are no questions. Okay, all right.
Well, actually, I have a question, Jahar. So in which way are the calcium signals received by the type 2 cells? So are there specific receptors that mediate the response to calcium signals that are not present in other nearby cells? And are those signals, have those signals been shown to be responsible for the ability of the type 2 cells to either secrete surfactants or respond to infection?
Right. So the initiation of the signal in any cell type, which is either the type 2 or in this case, the macrophage, is usually a receptor-mediated process. So I'm thinking, well, the spike protein might engage ACE2 and generate a calcium spike in the cell which it is interacting with. Now, the calcium now goes from one cell to another in the lung through gap junctions. We know that these cells are all connected intensely with gap junctional communication, to the extent that all of the epithelium and the macrophages of the lung are like a syncytium. And so what happens is that you get infection. Even if you get infection in one alveolus, multiple alveoli around that alveolus also sense the inflammation. So presumably this is a defense mechanism to augment the inflammatory response. But of course, if this is an injurious situation, it also amplifies the injury. So we think this communication might be a mechanism for the severity of the disease here, because the inflammation is so fulminant.
So Jahar, we have a question from Richard Axel about the role of the ACE2 in the endothelial cells in the context of this process.
Yeah, I think that's a very good question, because the endothelial cell is right next to the alveolus, and there's a very small barrier between them. But the barrier is extremely tight and does not let anything through. So the question is, once the virus gets into the alveolar space and infects alveolar cells, how does the signal go to the endothelium? This is something I'm not clear about at all, and I'd be very curious to know. It clearly could just break through in some mechanistic manner, but I don't know that. So it's not clear to me how the spike protein of the virus would access the ACE2 receptor on the endothelium, but it's well known that the ACE2, that receptor is expressed on the endothelium. And so we are able, with our preparation, to actually address that question. So indeed, if there's viremia, that could be a scenario where the endothelial ACE2 is involved. We will certainly test that idea.